Assalamu alaikum warahmatullahi wabarakatuh. My dear children, this is a continuity of our discussion on unit number 14. We are at this place by completing page number 8. Today we have to come to page number 9. Okay, if you consider page number 9, I have totally worked on tissue culture on this lesson. Okay, so I am going to discuss tissue culture to you with this. So actually with the page number 10, vegetative propagation, that means sexual is natural, artificial, all the vegetative propagations are ending. So in that case, what is tissue culture? What we will be getting, that is the idea I am going to discuss here. In tissue culture, my children, the very first thing it is, we prepare a culture medium. Okay, we prepare a culture medium in a petri dish. You can use anything to do with, but the petri dish I am using here. On this petri dish, culture medium, you are going to place a small plant tissue. This plant tissue you are placing is called explant. So you are placing an explant on a culture medium. Okay. On a culture medium, you are placing an explant. After a couple of days, what you will see in the culture medium is from the explant, okay, a green color mass will get created. This mass, what is getting created, is called callus. After few days, in the callus, okay, now in the callus, plants, kuti plants, we used to call them plantlets, will start to develop. Their roots will develop, their stems will develop, like that. After a certain size of growth, we separate them. And transfer them to a separate medium like this. Individually, there are 4, 5 mean, 1, 1, 1, 1, like that. After a certain amount of growth, we will transfer the plants to a tray of sterile soil. Right? Now the plants are planted in, what's it? The sterile soil. Very clean soil it is. After they get adopted. Remember, this is culture medium. This is culture medium. Okay. Now only they are transferred to the soil. After certain length of growth there, they will be planted in the ground. Okay. They will be planted in the ground. Now they are brought to the field. Okay. Now they are in the field. So this is how we are conducting tissue culture. In the tissue culture, you have to draw this diagram and mark the details. Okay. In the tissue culture, what is tissue culture is mentioned to you here. Production of new offsprings which are identical to the parental plant by cultivating any vegetative tissue of a plant in a culture medium under controlled conditions. Okay. In vegetative reproduction, you know that one, all the organisms, all the offsprings comes are same. When they are coming same, we are describing them by the word called clone. Right? And thereafter, you know, in the beginning, there's a small plant tissue we are introducing to the culture medium, and that is called explant. Okay, that is called explant. It is the beginning part of it. According to your syllabus, apical buds, lateral buds, root tips, those ones can be used as explant. According to my experience, not only them, leaf pieces, flower petal pieces, even them also can be used. You know, there is a culture medium here. Okay, one of the most important part here is there is a culture medium, right? That culture medium, when you consider, it is a tissue culture medium. I will show you here. It's a jelly-like medium. It's a jelly-like medium. You can see here, this white color component, what you notice is the culture medium. You can see the complete plantlet. Kuti plants are called plantlets. Those plantlets have grown in the culture medium. If you consider the culture medium, in the culture medium, there will be various ingredients. There will be sucrose. There will be minerals, salts, vitamins, plant growth substances, and there will be a carbohydrate called agar to make that medium solid. Okay. Further, we keep this. Now, if you visit a tissue culture laboratory, it's a very clean laboratory. Everyone can't go inside the tissue culture laboratory. That much a very clean laboratory, the tissue culture laboratory. That's why they are telling sterilized conditions. It's an air-conditioned room. It is working under controlled temperature. No very bright light or very dim light is used. It's an exact light intensity is used. That much a controlled condition, a tissue culture lab. Okay, I will show you here a tissue culture lab. Okay, there's nothing to see much in a tissue culture lab. Okay, just for your idea, I'm showing you the tissue culture lab. Okay, now this kind of uniforms are there for them. Even a single hair should not fall. Okay, that kind of an environment is the tissue culture lab. Okay, you can see the plantlets have grown in different stages, right? You can see there's a cool light pattern there. It's not so brighter, it's not so now you can see here the plant has grown in the culture medium, right? You can produce millions of plants in a shorter space like this. That is the biggest benefit of it. Okay, right. And what are the principles? I mean that what I explained you. Now I explained you know the diagrammatical, this is what we do, this is what we do, this is what we do. And I have used a word called callus. Okay, for you to, I want you to see the callus. Okay, callus in tissue culture. I'm searching callus in tissue culture because there's a disease also called callus. Okay, this is the appearance of callus. You can see the callus. Okay, it is light green in color. Okay, a sweet meat like. Can you see? Okay, this is called callus. You can see small plantlets have grown out of the callus. That's how they grow up. It is an undifferentiated cell mass called callus. Okay, fine. So your book is telling it's an undifferentiated cell mass. From that one, new plants can, new roots, new buds, new leaves can grow out. Okay, and once you go for the tissue culture advantages, almost vegetative propagation advantages, but some new benefits are there. Production of offsprings that are identical to the mother plant. This can be seen in all the vegetative reproduction. Second one, large number of plants can be produced at the same time. Now, one day I visited to a farming school, and in that farming school, two women were conducting grafting, vegetative propagation. So I was interviewing them. Amma, per day, how many plants you would be grafting? Then they told, Udayindang, Vedakalot, Mahatya api desiyak vitrahadan. We will be making around 200. So per day 200 mean monthly 6,000 maximum. But if you give the time to tissue culture, you can make millions in a month. That is the biggest benefit that. And the time taken to make them is also relatively short. Production of large number of plants in a short period of time. Propagation of large number of healthy plants, very sterilized condition, healthy plants we are making in a small space. Tissue culture lab will not be very bigger. Okay, and you can produce large number of plants using hybrid tissues with favorable genes. In unit number 20, inshallah, I will discuss you regarding the hybrid tissues. Hybrid plants are a variety of genetically modified plants we are getting, okay? We benefit of them. And finally, the advantages of vegetative propagation. One, ability to propagate plants that do not produce seeds successfully. Ability of having offsprings that are identical to the mother plant. Propagation of plants that bear fruits easily. Ability to propagate selected plants which are resistant to diseases and pests. And ability of generating plant varieties which withstand harsh environmental conditions. The basic thing is this. If you find some good quality, that good quality can be preserved by vegetative propagation. It can be resistant to diseases, it can be resistant to pests, it can be withstanding harsh environments. That character will not be allowed to change by vegetative propagation. Sexual propagation is not so. Okay, asexual propagation will not bring variations. Sexual reproduction will bring variations. Variations are important for evolution. So vegetative propagation will not contribute to evolution. because they don't bring variations, no differences. Okay, no differences. If differences are made only, evolution will be conducted. In a family, if all the children are 100% same to each other, will they reach different heights in their life? Okay, they are reaching different heights in their life mean they are reaching different heights there in the life mean they are different. That is called evolution. If all become doctors, who are going to be the patients? That will be a hypothetical question. Okay, so up to page number 10, I have completed everything. So this is the note you have to write. Okay, tissue culture, explant, culture mediums, constituents, conditions, then principal steps in tissue culture. Thereafter, advantages of tissue culture. Thereafter, advantages of vegetative propagation, and ultimately disadvantage of vegetative propagation. Okay, inshallah, I'll meet you again with a new video to further proceed sexual reproduction in plants. The next coming 11 pages are going to be based on the sexual reproduction in plants. Good luck and good night. Assalamu alaikum warahmatullahi.